What is Ebola and Zika virus? Ebola virus is a rare and deadly disease caused by the Ebola virus, while Zika virus is a single-stranded RNA arbovirus within the flavivirus genus. How is it transmitted? EBV can be transmitted through zoonotic introduction or through close contact with infected blood, secretions, and other bodily fluid. ZIKV is primarily affected by infected datum-biting females Aedes aegypti and Aedes albopictus mosquitoes.
What is pathogen surveillance system? It is a systematic collection, analysis, interpretation, and dissemination of information regarding the occurrence of disease due to the specific pathogen for public health action to reduce morbidity and mortality. There are 4 types of pathogen surveillance systems, which are genome sequencing, in-transmission focused investigations, epidemic investigations, and digital epidemiology. The first one is genome sequencing. For sequencing the genome, normally we use next generation sequencing platform, but the equipment is mostly benchtop and the sequencing need to be done from the culture. The characteristics are, can be easily shipped, operate under rough field condition, and small. The example of portable sequencer is the MinION. It has pocket-sized features, can be powered through a laptop USB connection, and had been used in field situations during the Ebola epidemic.
The second type is in-transmission focused investigation. It uses genetic variants to identify person-to-person transmission events either through manual interpretation of the variant shared between outbreak cases or via model-based approaches. The steps are: first, the outbreak isolates and non-outbreak isolates were sequenced and their genomes are either assembled de novo or mapped against a reference genomes, and the genomic differences between the sequences are identified. The second step is, these features are examined to infer the relationships between the isolates from whence they came. The last step is, genomic evidence for epidemiological linkages is reviewed in the context of known epidemiological information. This is the overall process. The third type is epidemic investigation. Only a subset of the epidemic cases were sequenced. The goal is to use population structure of the pathogen to understand the overall dynamics, uses phylogenetic approaches that captures both epidemiological and evolutionary information from measurably evolving pathogens. Example of an phylogenetic tool is BEAST. Sequence data are used to build a time-label phylogenetic tree using a specific evolutionary process as guide. Helps in inferring epidemiological parameters including the basic reproductive number RO. Next is digital epidemiology. It is a modern surveillance system using human, animal, environmental, and other data to carry out disease-specific surveillance in which a single disease is monitored through one or more data streams. The steps are, first, information is retrieved from a range of sources including digital media and newswires. The information were translated and processed including extracting disease events and ensuring reports are not duplicated. The trends were analyzed and the information was spread to the community through media.
Next, a future model for surveillance and early outbreak, 2027. This is the overall flow of the process. Step 1. Syndromic surveillance system reports unusual trend of purchasing medication in a specific area. It will analyze the specific area and find the pathogen spreader. Step 2. Alert is triggered. The field response teams meet the citizens. Nasal swabs are taken from the human and livestock with fever and also from the pathogen spreader. After that, samples are immediately analyzed using portable DNA sequencer. Step 3. An app on phone reports and reveals the pathogen involved. The data release triggers an announcement via social media. Within a minute, interested virologists create a shared online workspace and open lab notebook. Implementation of pathogen surveillance system in Malaysia. The surveillance system is used for respiratory and diarrheal pathogens at human pig interface in Sarawak. Large livestock operation and dense human population of Southeast Asia are considered a hotspot for emerging viruses. Viruses such as PCV2 aerosolize at the animal interface, and human walking in this environment carries these viruses in the nasal airways. Pig feces, pig oral secretion, bioaerosol, and worker nasal wash samples were collected and analyzed via RPCR and RRT-PCR. These are the steps involved. Nucleic acid from a number of targeted viruses were present in pig oral secretion and pig fecal sample, and that several viruses were detected in bioaerosol sample or in the nasal passages of humans with occupational exposure to pigs. As for the shortcomings, the study sample did not provide robust data for a confident risk factor analysis, especially among the human workers. These are our references.
Thank you.
Arigato. Kamsahamnida.